Okay, let's now look at the visual pathways. The visual pathway. Okay. I would like class for you to look at this video here. I think it gives a nice video on the visual pathway. But anyway, let's get started. Okay. So right here would be, this is a rod, or or if it was a cone. We're starting at the very beginning. Light would be perceived here, then everything we've discussed in a previous video, till finally I'm at the ganglion cell. The axons of the ganglion cell, which do action potentials, everything before that was graded, action potentials then come in and form the optic nerve. All right, so we're going to go through this. All right, here would be the optic nerve from this eye and the optic nerve from this eye. Then the optic nerve comes down here from that, and I want you to notice that the fibers right here from this lateral aspect of the eye go into the optic nerve, but they go on the same side, coming into this optic tract right here. And these go on the same side. Now a crossover occurs here, if you'll notice, right there on the medial aspect, a crossover occurs at something called the optic chiasmata. The optic chiasmata. Okay? So here's that optic chiasmata in an actual cadaver brain, optic nerve. And so when you look at this, the lateral aspect of the eye is going to be on the same side going into the brain area here, that lateral aspect, but the medial crosses over. So when you look at an optic tract, then it has some activity from both eyes. Some activity from both eyes as we come down because of this. All right. Now, I drew these arrows here to show you this, that each optic tract right here looks at a different eye field, should we say. Okay, this optic tract here then, which would be the blue, would be this. This eye right here is looking here on this right side. That's the temporal aspect of this left eye. But the medial aspect of the right eye is looking there. But if you look at it, both of these are in the same optic tract. So that means this optic tract is looking at this right eye field here. Now I put these in red. That means vice versa here. This one is looking at the left eye. So the right eye temporal aspect looking at left, and left eye medial aspect looking at left. When you get a chance, you can look at that. Okay. So we come down here. Here is this optic tract here. This was the optic nerve, and here's the optic tract. Then fibers from the optic tract are going to synapse on neurons in the lateral geniculate nucleus of the thalamus. Okay. So then once that synapse occurs, then this carries into the optic radiation, which will take us into the occipital lobe of the brain, into what we call the primary occipital cortex. Vice versa on this side. Okay. Some fibers here are going to go to what we call a pretectal nucleus right there. So some are going to go here versus going to the lateral geniculate nucleus. This is in the midbrain. Okay. Then some fibers are going to go to something called, if I can see it here, the superior colliculus, which you don't see on the drawing, but well, yes you do, in the superior colliculus right here. So we'll be looking at that, and then for the optic radiation to the primary occipital cortex. So let's go back again just to be sure we're all on the same page. We start out with the ganglion cell axon. From that forms the optic nerve. The optic nerves come in, and some of the fibers cross over in the optic chiasmata. When they cross, the minute we get past the optic chiasmata, we're into the optic tract. Then some of the fibers of the optic tract synapse on the lateral genicular nucleus in the thalamus and take us into the occipital lobe of the brain. But some are going to synapse on the pretectal nucleus, we'll talk about, and some on the superior colliculi right there. So that just gives a quick run through of what we're looking at there. All right, so let's go further now. So the axons of the retinal ganglion cells, okay, from the nerve, those ganglion cells we talked about from the nerve, medial fibers are decussate, crossover means at the optic chiasma. Most fibers on optic tract continue lateral genicular of thalamus. Okay, so we just talked about that. The lateral genicular nucleus receives information. Now this is part of the thalamus. Here's your thalamus here. The thalamus has several nuclei. Remember what a nuclei, a cluster of neuron cell bodies in the central nervous system. So it has several nuclei. Right here is the lateral genicular nucleus, right there. So that's what we're talking about. So I go back. So continue to the lateral genicular nucleus, which we just spoke of. There's the thalamus. Lateral genicular, and the lateral genicular nucleus receives information directly from the ascending retinal ganglion that we talked about via the optic tract and also from the reticular activating system. Now the reticular activating system is a diffuse set of neurons that kind of keeps the lights on in the brain, I say. In other words, the firing frequency of the reticular activating system determines your states of consciousness. So it keeps the area of the brain alert. That means the firing frequency goes down when you go to sleep. Neurons of the send axons through the optic radiation, a direct pathway to the primary visual cortex. Okay, and in addition, the cortex sends some fibers as far as on a feedback mechanism. Okay, we go further, and there's the thalamus. The fibers from the thalamic nucleus form optic radiation. Optic radiation connect to primary visual cortex, which is in the back. But some fibers are going to go to the superior colliculi. Okay, the superior colliculi is a member of what we call the corpora quadrigemini, which is located in the midbrain. There's four bodies there. The superior colliculi 2 and the inferior colliculi 2. The superior is dealing with vision, the inferior is dealing with auditory activity. Okay? Right here is in the midbrain that you see. Okay? And I'm going here. The roof of the aqueduct, cerebral aqueduct, has the corpora quadrigemini. The two superior colliculi control reflex movements of the eyes, head, neck, and so forth. Let me tell you what that basically means is that if you're looking at an object and you want to follow it, then your eye muscles, you remember the extraocular eye muscles, superior oblique, lateral rectus, so forth, would move with you to follow this moving car, this running person. In addition, your head and neck would turn. So this coordinates the head, neck, and extraocular muscles as you try to follow something with your eyes. That's what the purpose of this is. All right. Now, a small subset of ganglion cells in the retina contain melanopsin, I should say, circadian pigment. It projects to the pretectal nuclei. The pretectal nuclei is also in the midbrain, and it deals with the pupillary reflex. Okay, you know, in an exam, neurological exam, we'll shine light in the eye, and we'd like to see both eyes. Even though I'm shining one eye, I want the other eye to also constrict the pupil. If it doesn't, then we know we have a problem somewhere in the brain. Okay. Some fibers Also from this subset going the suprachiasmatic nucleus. That's a nucleus located above the optic chiasmata, and it deals with circadian rhythm, sleep-wake cycle, sleep-wake cycle. So that's what you're looking at there. All right. So we go a little further here. Here again is that picture we looked at already. So we have the primary visual cortex, which is back here. So let's go here. Okay, there's the thalamus. There's that. Okay. We have this primary visual cortex, which is back here. And then we have the visual association cortex. Okay. The primary visual cortex, in the extreme posterior of the occipital lobe, is involved in visual information. In other words, that's where you would see from. But the visual association area, which is this one right here, surrounds the primary visual cortex and interprets visual stimuli like color, form, movement, things of this nature, to put meaning to it. We'll speak of that again in just a second there. Okay. So I come here then, here would be a picture, that's that occipital lobe again. Okay. So now we're looking at this aspect of depth perception. Depth perception is going to involve the use of both eyes. Okay, in throwing a basketball to hit the hoop, you have to have a three-dimensional image. You have to be able to determine depth perception, how far does the basketball go from you, to make it simple. So what it involves is confluence of both eyes. Some things are overlapping from one eye field, another eye field, and some are not. But it requires input for both eyes. Now, we are binocular, so we have both eyes. If I look at a lizard or some other animals, what will happen is their eyes on kind of on the side of the head, shall I say. Their thing is to get away from enemies, things that would attack it. We, on the other hand, concentrate our eyes to get more of a three-dimensional image, where it's more difficult with animals that have on either side of the head. Okay, and you can read on that. Now we're looking at visual processing. Retinal cells split into channels, each with its own type of ganglion cell. Okay, so these channels involve, remember when we were talking about the visual association area as to how it would look at color, intensity, and so forth. These channels, so they're special ganglion cells, should we say, and these channels will synapse on that, and some of the channels, ganglion cells associated with the channels, will look at color, brightness, angle, direction, speed of movement, things of this nature there. Now, we also have to have sharpness of image. The sharpness of image, I'm going to go back to this, go to this picture here. That's where things are turned on and off, are controlled on what we call lateral inhibition. And lateral inhibition, if you can see, the amacrine cells and horizontal cells connect to several of these structures in the retina to kind of give a sharpness of image, turning on some, turning off others to give that sharpness of image that you would see. Okay? All right. We go a little further here. So the lateral genicular thalamus process depth perception, cone input, primary visual cortex we talked about, cortical processing. Now, the processing involves several things. Okay, though back here is the eye area, and this is the visual association area, you still are going to send signals to the frontal lobe, so forth like that. Okay, so I'm looking at a car. I would see it here. I would get some indication of its red, blue, green, whatever, and its shape. But my interpretation that that's a car would not be back here. The interpretation that's a car would be located in storage within frontal cortex areas and areas of the hippocampus and other areas there. So there are multiple